first things first, it's important for us to recognize our sponsors. So the Minnesota Soybean Research and Promotion Council funded this program this year. So we want to thank you for your checkoff dollars. Minnesota Wheat and their promotion council, they provided their most precious resource, which was Missy Carlson and Chris Matter at times, and so we sure appreciate that. Agaseed Seed and Supply, they're a cover crop seed dealer that provided some cover crop seed for us. Minnesota Ag Services is a gentleman, Dorian Gatchell. He worked for the program. Bayer Crop Science, in a year where there were worries about glyphosate scarcity, they were kind enough to provide some to us for this program. Obviously, University of Minnesota Extension. We'll get and show you a little bit more about our trials. Before we start, I just want to let you know that we do have some small plot trials as well. One is at the Northwest Research Center in Crookston, and the other is at the West Central Research Center in Morris. There we are looking at different and more treatments. So at the research centers, we're looking at tillage, we're looking at seeding rate, cover crop seeding rate, and then cover crop termination timing, and we're looking at more things. So we're following nutrient cycling in the soil, we're looking at soil health parameters, IDC scores in soybean. We're looking at weed management in soybean as well. And so unfortunately, we don't have that data here to share with you today. What we are sharing with you, though, seeing as this is an on-farm research summit, is the results of 5 different on-farm locations. So in 2021, we planned to have at least 3 locations kind of around that Crookston Research Center and then around the Morris Research Center. So these were farm-sized plots. The farmers used their own equipment similar to the Minnesota Wheat On-Farm Research Network protocol. Up north, we had a trial in Gently. The blue dot there is showing Barrett. We have a plot at Tinta, which is orange. The green dot is Browns Valley, right there at the part of Minnesota that sticks out. We also have a location at Appleton, our southernmost location. So at each of these farmer locations, we had 4 different treatments. These treatments were randomized and replicated 3 times. We had a no cover crop control, where the farmer just did whatever he planned on doing as far as tillage is concerned. The other 3 treatments had cover crops. They were terminated, though, at different times in relation to soybean planting. So we had a termination. The first termination timing was 1 to 2 weeks before soybean planting, at soybean planting, and then we had 1 to 2 weeks after soybean planting. So on farm, we had 2 fields where 40 pounds of rye were drilled last fall. We also had 3 fields where we had a drone actually seed the trials. So here's a video of the drone. So in those, because we were broadcasting seed, we bumped up the seeding rate to 56 bushels, pounds to the acre, and then did that into standing corn. In Gently, the previous crop was wheat, and down in the west central locations, the previous crop had been either corn grown for grain or for silage. One of the things that we looked at, we need to know how much rye we actually were able to grow in these plots. So we took this fancy scientific tool, it's a hula hoop bought from Walmart, and tossed it into the plots and collected all of the biomass.
Next year, we're going to pick smaller hula hoops.
live and learn. But you can see on the left picture there, that's before somebody took a scissors and cut all the rye out of there. And then on the right, that's afterwards. So just the rye was looked at. So we just collected the rye there. We separated out the weeds, and we didn't allow any of the previous crop residue to get in those samples. Many of those samples were brought to the research center in Crookston. Peyton Loss here handled quite a few of them. They were dried and then weighed to get at our biomass. So how much rye was collected per acre? So just to kind of get your mind around it, I need to think about things in this way myself. So one large square bale is about 600 pounds. And in these various plots, we collected between 1/4 of one of these bales and almost up to 8. One hula hoop at a time, you know. Per acre, yes. Peyton didn't have to deal with eight bales worth of biomass, luckily. Jody.
Yeah, so Appleton is a irrigated sandier soil, and so he was also ridge till. So we try to go with people who, had different types of tillage systems out there. And I think he might be the last ridge tiller out there. Might be 3. We drone applied the rye the fall before, and this year we have a new drone, which is good. It has a bigger tank. The tank, last year we had to go over the plots twice. The reason why we didn't go with an airplane is because we had plots in there that had no rye at all, and we didn't, the plane is not that accurate, so we had to do it with a drone. I would not say right now that's very economic. It was $150 an hour, so yeah. The rye did not establish as well because of ridge till. He has a ridge and the residue kind of goes in between the rows, and it was really thick residue, and the rye did not establish in those areas. It established more on top of the hills. And it was the earliest planted field. Actually, he called that morning and says, "I'm planting today." And we're like, oh, because we're supposed to have the treatment where we killed the rye, a week to 2 weeks ahead of time. That one didn't happen. So what we did is, instead of having the termination 1, he let us have those plots go an extra month further out. And we don't have that data on here, but yeah, you can have too much residue, we found out. And if you know me, that's saying something, because I love residue. So here's Appleton. Again, look at T3 is on the left, and we only collected 384 pounds per acre worth of residue, or worth of rye biomass there. T2 is at planting. That was on May 10th. That was 146 pounds. This is the lowest field of residue, or of rye biomass. Again, looking at the ridge till, you can see how much residue is out there. It was very thick. And then the one where he let us go another almost 40 days after planting, you can see that the residue is still, or the rye is still standing, and it never went to the ground to be decomposed. And so other people are saying, well, could you roll those plots? Yes, but we were trying to keep all of these fields the same, and so we did not roll on these ones. When you start seeing the statistics coming up, you're going to see that one of them had a 10 bushel difference in yield. And we're going to say that wasn't statistically significant. And you're going to say, yeah, but it's economical. It's not. And the reason why is because this is, how variable are your fields anyway? This is a yield map. This is showing a field that they did the same fertilizer, the same hybrids, the same everything, same planting date, everything was the same out there. And this is the variability that they're getting. And you can see with the corn that you can have anywhere, 60 bushel difference. With wheat, you can see 20 bushel difference. And in the combine, you've seen even more extreme than that. So what statistics does, so if we put our trial in the middle of that, what's making the difference? The natural variability out there, or what we actually put out there? So stats takes out that background noise, helps just take it out so that you are just looking at what your treatments are doing out in the field. And if there's a lot of variability, those numbers can be much different. Your yields could be 10 or 10 bushels apart because of just the natural variability out there. Some studies are really tight where you can see a bushel and a half is significant. Well, that means that the field wasn't that variable. That's why small plots are something that we like to start on, because it takes out a bunch of this variability and gives us just a few things to look at so we can really hone it in. And so next is going to be the yields, and you can see what I'm talking about. That field was kind of variable. Oh, and to set it up, it was all a loamy soil, and it was very consistently just one series out there. But he's irrigated, and he did have to run it. Now, was the sprinkle, did it irrigate evenly on all the plots? We're not sure. So that's the thing about field trials too, is there's a little bit more noise out there.
Let me explain this particular graph because we're going to show you 5 of them. So on the bottom, that's just the treatments. Usually we're starting in the left-hand side with the no-rye treatment, and then we're going with time from the before planting, at planting, and after planting. But if you'll recall, for this particular field, we didn't have a before planting treatment. The bars themselves are the yields. The numbers, the red numbers above the bars, those are the actual yield data. If you take a look at that y-axis, here. On the left-hand side, it's showing yield. On the right-hand side, we're showing biomass, and the biomass is this little line right here, and the values, biomass values in pounds per acre are listed right next to it. Most of the treatments had significant differences among the treatments for biomass. We do have tables in your book. It's the last set of studies that are detailed in your on-farm research book. If you want to look at that, we also have soybean stand count data in there, which I think can really go a lot towards explaining some of what we're seeing here as far as yields are concerned. But you'll see here, and this is the one where Jody was talking about almost a 10 bushel yield difference. That's a numerical trend. Statistics didn't tell us that indeed that these treatments were statistically different from one another. Complicating factors. So in each of these trials, we're going to show you the data and then talk about some of the factors that added variability. Here, there was seed corn maggot issues, bean leaf beetle injury, brown stem rot. We also had some herbicide carryover injury from acetochlor. And then Jody has a video here of the ridge tiller system actually chopping up the rye itself.
He was freshening his berm. So luckily we were out there to take the samples because it cut the rye and pushed it to the side, and it was like, oh, we need that for biomass. So we were picking up little pieces and putting them in the bag. Yeah, so this was a different system. Each of these systems we kind of got to explain because every farmer did something a little different. So in Browns Valley, that's more of a clay loam soil. He was no-till. He also had cows out on this field afterwards. During the winter, so the rye had come up. So last year we lost one of the fields up here, mainly because it was so dry and the farmer's like, it's not going to germinate. And then we got a whole bunch of rain. And so all the other fields did germinate, which was great, and the cattle enjoyed it as much too. The corn was a lot shorter, and so it let through sunlight, so he got a really good catch. His rye really took off. And we'll talk about some of the issues with cattle. I don't think we're going to do any more ones with cattle. They don't poop where you want them to.
Although it's nice to be able to actually drill into the cover crop into those fields, yes, that is definitely a complicating factor.
Okay, so here we're looking at, he had no-till, no rye, and you can see he had a lot of residue there too, but the other thing you can see in there, the stuff that's starting to die out, is alfalfa. And alfalfa was in there 2 years previous. So it was alfalfa, corn, and then soybeans, and the alfalfa was still coming through. That's also cows like that as well. And then you can see he had about 1,280 pounds per acre, so a couple of straw bales out there worth of rye residue, and then at planting it was about 3,000 pounds, and then afterwards even more. So it was significant how much more rye he had at each termination timing. And you can see for the T3 up there, you know, where we terminated at 10 days after planting, how much rye is up there. It's, yeah, I knew we were going to be in a little trouble with that one. But oh, and we also had alfalfa issues that would just pop up in a certain area. So this is an aerial view. You can see the rye coming up through on the T3. It's not, we need to be a little darker here, but on the T3 ones, those are the ones that have higher rye residue in there. And it showed up all year. It was left standing. It did not go to the ground and decompose. We missed that spot. We went over it where the alfalfa affected it. But otherwise, these are all done by combines and grain carts. And then also looking at their yield maps too.
So here's the yield data. So the no rye treatment, Jody talked about the biomass, so I won't talk about that here. Those letters on the bottom of each of the bars shows us the statistics. Those treatments with the same letter are statistically similar. So you can see that the no rye before planting and at soybean planting treatments were statistically similar. So they didn't differ from one another. It was only when we got to that after planting treatment with the 4,600 pounds of rye that never did lay down. If you take a look at that picture on the right there, that's just showing the rye just kind of, it had reached reproductive growth stages and it just wasn't budging without rolling it or something like that. And so that definitely had a significant effect on yield there, as you can see. So complicating factors, Jody captured a cow patty. So that's the picture you're looking at right there on the right. One complicating factor with the cows was that all of those cow patty areas tended to have more weeds. And so there were potential weed issues that might differ between the plots. We haven't analyzed that data just yet.
Yeah, when we were out there, you know, cattle over winter isn't going to go into the no-till plots, right? There's not as much to eat. They're going to go in our rye plots, and that's where they plopped in our plots. And they carry a lot of weed seed in their manure, and so we were finding a lot more weeds in the rye plots, and we're like, well, that shouldn't be that way, but it was the cattle. So then in Tinta, we're up at a silty clay loam. Tinta is also in Traverse County up at the north end of it. The previous crop was silage, and so he was able to drill in the rye. So he's not going to have the residue, you know, that the other plots did because he was silage and took most of it off. So he went with no-till, and then there was no seed treatments used here. This is what it looks like when you have no-till and no rye. And then you go to 22 days before planting. See, the rye did grow, but it lays back down with planting, and you know, it wasn't or it was only in its vegetative state. And then when you go to planting, we had a lot of residue out there. And then even after planting, terminating that rye, both of those plots were very obvious from the road and from the air, and that rye never laid down. So the problem with that is it was shading the soybeans as they're trying to grow.
An additional issue we saw at Tinta was that there was some root rot issues. So this farmer didn't use a fungicidal seed treatment. And so you can see it's particularly these kind of round patches right here. Those are areas where there were root rot issues in the soybeans. We did get them analyzed, and several things were found. So of all things, charcoal rot was found. I know it was a pretty dry year in this location, and so charcoal rot isn't It's not that surprising, I guess, that we found the charcoal rot pathogen, but probably much more responsible are Fusarium species. So we're not really exactly sure which species, but it could even be the species that causes sudden death syndrome in soybean, because they produce a kind of a bluish color. The spore masses have a bluish tint to them, and that's what was seen. One thing that I just want to mention here is that at these on-farm trials, we've applied for an MDA Agri grant. And so the goal of this grant is to be able to use these on-farm locations to see if using a cover crop and maybe cover crop termination timing has an effect on soybean disease as well. We're hoping to get funded there, because then we would have a student to do some of the work as well. But here's the yield data. You can see again a lot of rye was produced. You can see also that the no rye and the before soybean planting termination timing were statistically similar, but the at planting had significantly lower yield than either of those no rye or before soybean planting plots. And then the after planting termination timing had lower yields still. And so you can see that between the no rye treatment and the after planting treatment, that's around 9 bushel difference. So complicating factors here. So here's kind of a close-up, a drone shot of some of the root rot issues in the particular field. This was our wettest location. It also was the latest planted. One thing of note is that there was so much rye residue in that after soybean planting termination timing that, you know, when you run the combine, all that rye has to cycle through the combine too if it's still standing up. And so that actually reduced harvest speed by 1 mile per hour.
Yeah, and he had a draper head on there. I mean, it was so obvious that we had to slow down because of all the rye that was coming through there. So that is another thing to take into consideration. So then we went to Barrett, and we were in a clay loam again. He did fall vertical till, and the previous crop was corn for grain, so there's going to be a lot of residue out there again. And then also, he found there that he had better trafficability with rye. So when we set up these plots, and then they need to go through with a sprayer, I don't like them, I want them to go perpendicular, which now you're never going to do research with me, right? And the reason why is because then you get the compaction evenly across all the plots, whereas if you use a sprayer in the plots that we were in, they're the size of their planter, and the sprayer could be, you wouldn't get the sprayer tracks in every plot. So I didn't want wheel traffic to affect the yield. We just want to look at the rye. So we found out that when he went across with the sprayer perpendicular to all the plots, where there was no rye, the sprayer actually sunk more. And and I had pictures of it, and for the life of me can't find them, but I will get them back somehow. It was pretty obvious. And then we also found in Tinta, it was kind of interesting. We went to go dig up plants for the diseases, and Angie's sitting there like putting all her weight, you know how dry it was, all her weight trying to get.
I was jumping on the shovel.
Jumping on it, trying to get the roots. And that was in the no rye plots. And you could see cracks in the soil. I mean, it's definitely dry, at least 3 feet down, because we couldn't even get our fall samples out, and fall soil samples. And then we went over to the rye plots, and you could get, it was still bone dry, still cracks everywhere, but you could get the shovel in. And it was like, I was kind of floored away by that.
Anecdotally, just how easily that shovel went into the soil I thought that was really personally impactful. I really, it was kind of eye-opening, personally.
What are we at next?
Barrett.
So this is what it looks like. You can see on the left-hand side that the residue is sized because he did use fall vertical till. And then left, Yeah, and then before planting, we had a lot of residue, because that's no-till, with a lot of rye, and the rye just got a lot more as he went across. Again, too much rye was out there, and we did have to slow down with the combine in this field too. And so that's what it looks like from the air, and you could see the different treatments, even the different colors of the beans throughout the whole season.
So here's the yield results from Barrett. You can see that all of the rye treatments tended to reduce yield here compared to the no rye treatment, regardless of when you terminated. And if you'll think back to some of our other locations where we had rye biomass that was like 4,600 pounds, this biomass accumulation was a lot less than at other locations, and yet it still seemed to have an impact here.
But he had the most amount of residue, corn residue, of any of the plots. So I think it was kind of just too much residue out there between the rye and the corn residue.
Again, lots of residue, one of the wetter fields.
And we seeded this one by drone, and you can tell it's not taking off in the areas that had a lot of residue. That's why it's in strips like that.
Yeah, this was aerially seeded, and so it's not as if we planted it this way.
So then we were at Gently, and he has, and Tim, you can add to this if you want, because he's here. Loam to loamy fine sand. He had a previous crop was wheat, and then he could drill it in, because wheat comes off early enough that we can drill in the rye and not have to do it with a drone or an airplane. And he did fall vertical till. So what they found, and Missy and Chris were, they were were invaluable, and taking all the data out there and really keeping up on this. And so you can see that we did increase on the residue levels out there. Did it go to yield? That was always the great thing. As soon as it went reproductive, we knew we were kind of in trouble. The other thing I want to say about this is, you remember how wet it was in the spring? So all this rye took off, and almost every farmer we worked with was planting the day before insurance went out. So everybody was almost a month late on planting, and that let the rye just take over. So this is not a normal year. What's nice is that we have the high end of residue from rye cover crop now, and maybe we'll have a normal year or a light year these next ones. What's normal?
Can I comment on that quick?
Yeah. Go.
Jody, you mentioned about the rye and the seed. Just to comment on the rye viability, it did go to head, and I think our final termination timing was right about the time the rye was actually flowering. But I was checking the heads all season. We didn't find there was some seed in the grain. I didn't find a lot of viable heads. It was seeds or the heads that went to seed, but when we were harvesting, we did know some rye seeds coming out in the grain. So that was something to notice, but just like Angie said, this is like the extreme of extremes here. Like, how often are we just starting soybean planting in the first week of June? Not very many years. So this, I would not expect to see this in many of the years if you were to try this.
Yeah, I would think that more normal would be your T1 is when we'd actually be terminating as our latest termination date, but not this year.
And we did want to keep the treatments, the termination treatments in relation to soybean planting. And so, yeah, the wet spring really kind of complicated matters. So here's the data from Gently. So you can see that surprisingly, the no rye treatment, the at planting and after planting termination treatments, those were all statistically similar to one another and higher than the before planting treatment. There was quite a bit of biomass out here, but there wasn't a trend where you had higher or lower yields with more biomass. And so we thought it through and came up with an explanation. So this is our best guess at this point. On the left-hand side is a picture of this field. That is volunteer wheat that grew last fall. You can see there's a lot of it out there. But come springtime, the picture on the right, that rye, or excuse me, volunteer wheat residue had broken down quite a bit. And so the theory here is that sometimes our soil microbes that do some of this breaking down of our crop residue, if they need nitrogen to be able to break down some of the organic materials. And so sometimes that can tie up nitrogen in the soil. And if that took place before the soybean nodules, rhizobium nodules had become functional, perhaps that's one explanation here. Oh. So some key findings. So we're considering this a unique series of experiments here. So we had a historic drought up here in the Northwest last year. This year in West Central, there was a drought towards the end of the growing season. Historically wet spring as well. So kind of the extremes. And so who knows how representative, even if we had another similar series of events take place, we're not sure just how representative it is. How much is enough? So we want our cover crops there to provide a lot of those ecosystem services. We want it to hang on to the soil. We want it to sequester some of the nutrients that remain in our fields. We hopefully want it to shade out some of our weed seeds that are trying to germinate. But we need to find that sweet spot where we have enough, but we're not negatively impacting our cash crop, because you guys aren't running a charity. So, and then another thing is, obviously we were doing this for research purposes, but if you can't terminate rye before it goes reproductive, try and knock it down in some way. If you can, roll it. Otherwise, you're going to be your crop's going to be fighting it all season long, getting shaded out. We do have funding for an additional several years, 3 years of data collection. So some of the farmers are helping us out again. So we have three locations centered around the Crookston Research Center, and then 3 locations around the Morris Research Center in West Central Minnesota. And so we'll be doing this all over again. We'll get more data. We'll be able to share more information with you, and we'll also have more challenges, I'm sure.
So we had a wonderful team that helped us out on this, because it's a lot of data collection. So we have the U of M people you can see there. That's from Crookston, St. Paul campus, and out at the counties. We had other awesome people like Missy and Chris, who honestly we couldn't have done this without them. They came down and helped us many times on just a quick phone call. So, and Dorian, and then our farmers, and definitely couldn't have done it without you guys. Wouldn't have a field. And the Soil and Waters did help out in the counties as well. And like I said, we couldn't have done it without them, because I'm getting old. So.
We all are.
It hurts your knees bending down that far for that long.
Better than the alternative.
So any questions on this? Yes, sir. He was asking about mechanical termination. No, we have not done any of that. It was all done with spraying. Yeah.
You haven't in past research either?
No, we haven't. We haven't had cover crop people on our team for a while, so now we do, and we can do that. But yeah, we have some. I know there are maybe on organic side that they've looked at rolling, but not with tillage. We definitely say don't try to till rye, because it just gets really angry. It comes back at you.
In your gently slide, with your volunteer wheat on wheat, did you do a biomass count on how much you collected? Because it looked like that was considerable in the fall.
Missy, we didn't.
Did we collect volunteer wheat biomass? Is that the question? We did not. That would have been good to know, but we did not.
Good question.
Biomass? No, thanks.
No, thanks.
With the, okay. With the sense of biomass, do you think it had to do with the shading, or was it the tie-up of nutrients or the lack of nitrogen or sulfur cycling?
We are doing the biomass nutrient levels. Those aren't all done yet. And we're doing the soil ones too. So Peyton, she's looking all excited. What did you have there? Did you have anything on the nutrient tie-up?
We did look at nutrients in the research sites, but those weren't presented up here. As far as the on-farm soils, those have not been analyzed yet. We did see some interesting for the research plots. And then for soil moisture and temperature, those are things that are looking at being implemented next year and in subsequent years.
Yeah, that sort of infrastructure requires funding, so we're trying to figure out a funding source there because it gets pricey, especially if you want to replicate it or at least have one series of monitors in each treatment at each location. Think about how much that adds up over time. But yes, ideally, we would have both soil moisture and temperature monitors as well.
That would have been
question.
great data.
I have a question. I've grown rye and it just volunteered after harvest. Is it possible that the rye gets too thick so that if you come in with a pea or a rapeseed, canola or type of cover crop, it would be too much volunteer rye that the broadleaves don't have a chance?
We did see, okay, so in the fields, we planted at almost 60 pounds an acre. And at the research and outreach centers, we used 20 and 40, but they were drilled. And what we found in Morris is that 20 pounds was too much. Our little soybeans were having a heck of a time trying to get through that cover. But again, this year, we were terminating rye when it was this tall and headed out. So it was just too much. And also pocket gophers really like to be protected by that rye. So we did find that, yeah, with the broadleafs, rye can be a little bit too competitive. And so we're trying to figure out what stage should we get it out of there. And I think it's going to be based more on the stage of the rye than it is where you are around planting time. Yes. Oh, you guys figured it.
Where you had these trials, we did this in Carrington, and we found that soil moisture can be an issue if you let it go to the reproductive stage, aggressively uses water.
Yeah.
And we don't have that much there. It's like,
Yeah, we
Ran out
did-
of time.
Yeah, we didn't get, we don't have all this stuff analyzed yet. I mean, we even have the weed biomass ones coming, but I know that the springs were extraordinarily wet, and then the tap was turned off for June and July and August. But do you have it for up here, Missy?
I just had another kind of a comment. In some of our other on-farm research that we've done with just rye versus no rye, like the site that we had in 2021 up in Roseau, saw specifically that problem. The rye, it wasn't even that big. Well, I shouldn't say that big. It was at about boot stage when it got terminated at soybean planting. But 2021, just the rain never turned on, so it used up too much water, and then it did cause a major water deficit in the soybeans that actually induced a potassium deficiency. So that can be, watching your soil moisture, yes, can be an issue.
Carrington, we found that we didn't have a yield loss or a yield penalty if we terminated at planting, but we always had decline after that because of the water.
Okay, I have a couple of comments and a question. We've done this several years in different scenarios with different crops. We've seen seeding rate of rye being very important, removal of the rye by grazing being very helpful, and also planting the rye in paired rows really seems to be a really good way to do it. That leaves you a space where there really isn't that rye competition where you can get your crop established, but yet you can still get the benefit of the rye between the rows. My question is about insects, and we've had crickets, grasshoppers in that rye, and it's caused stand loss. Is there any, have you looked at that and seen any?
You know, I don't think we saw issues with that this year
No, not
at
this
any
year.
of
I
our
mean,
locations.
found a few
except
stink
for-
bugs, but you were looking more at the disease aspect of it.
Yeah, I think our Appleton location was the only location where we had insect issues, and that was bean leaf beetle and seed corn maggot. But army worms are another thing that I worry about. You know, we also worried if we get our cover crop, our small grains cover crop seeded too early, and there's tons of cover crop acres around, are we going to be ending up with a hessian fly problem if we have wheat nearby in the springtime? You know, a lot of these things we just haven't found. This is 1 year, but I expect we'll be seeing all kinds of additional complications in future years.
Yeah, we've probably done this maybe 7 years now, so we're starting to see a lot of crickets build up in the spring. We're seeing probably slugs. It's just a lot.
I was worried about
interesting-
slugs
one,
because it was
yeah.
so wet.
A lot of interest. Yeah, it was a problem this year.
And what you're saying about drilling it in and only using a few rows instead of the whole thing, I do like that. And I also like the idea of guys doing this with strip till, where you put on the tank and you have it go on the outside of the berm, you know, the berming discs. So then you make a nice black strip there and you put the rye on the outside. And farmers are doing that too now. And I think that's a great way to give you more time to get that rye out of there without it affecting the crop.
You have a question, Tim?
Oh, yeah, Angie, Jody, thank you for presenting this. Now, my comment, you know, 2022 certainly was a unique year, but I don't want you to be overly dismissive of your results, because if there ever was a spring when having a cover crop out there to use up water and get you into the field faster was important, it would be this past year.
Good point, yeah.
Now, the second part, it comes down to the real question. Now, when the farmers went out to seed them, were all the treatments seeded on the same day?
Yes.
Oh, the treatments. Yeah, the rye was
did,
all done
yes.
within 24 hours.
Right, so all the soybeans were planted on that. So I suspect they probably waited until that control treatment was ready to go out there. So there would have probably been some days when they could have got out to the rye treatment sooner and got those soybeans planted. I think that's an important thing to recognize.
Yeah, that the rye could have sucked up some water and given more trafficability to get out there earlier than, yeah, definitely.
And thus far, we haven't, that's always something we've wanted to be able to do, is actually plant the treatments, you know, cover crop treatments when they're ready. It's a matter of finding the producer who's willing to do that.
Tons of spare time in the spring.
All right, we'll take one more quick question, and then I think we'll be able to, we're gonna move, we'll take one question, we'll move on real quick, but we'll be able to regroup this question in the cover crop panel too. But we should keep moving so we have time for our panel farmers to speak as well.
I would just like to add to the Carrington researcher, we do, at the Crookston ROC, we had this research, the smaller plots. And I can say that our 2022 growing season, we had an above average precipitation of 16.5 inches. I don't know what our overall like 5, 10 year, 30 year average was, but I just know that at 16.5, it was above average. And where was I going with that? I guess I don't exactly know. Train left the station. Yes, I would just like to add, because you asked about the precipitation in 2022, and that all did come in the spring, and like Jody touched on, it did stop in July.
And here's the one at Morris. With the 20 pounds, the 40 pounds, our population counts went down. Our yields ranged from 9 bushel to 69 bushels out in these fields. So you can have too much residue. So this is what, you know, it was horrible. Everything caught, everything grew, and it grew really well. So yeah, I think this is great that we're going to have the upper end of residue and rye residue, and now if we can get the other end of the scale to see where's that sweet spot to terminate. But again, if it's dry, terminate early. Okay. We'll be around all day.
So I also wanted to recognize Mike Lyseth. He is the plots coordinator at the research center, so in Crookston. And so Michael has done a lot for this series of trials as well.